TikTok videos are hilarious. All right, so today we review. So we'll review specifics about what you can expect to see on your exam, which is sort of standard operating procedure. 50 questions, multiple choice. And some of them I'll just tell you. So we'll start with that area. So you're gonna have to label the sarcomere. And there are 5 structures that you need to be able to label on the sarcomere. Still write multiple choice questions. But you'll have a diagram on your exam that looks like the diagram to every text or whatever you've got with the different parts of the sarcomere, and you'll have to label those things. Alright, so things you can do to label. Z-disc. The M line. This area between the thin filaments here is the H zone. Anywhere there's a thick filament, so from there to there, is an A band. Where there's not a thick filament, an I band. So while it's still multiple choice / matching, those 5 questions are on your exam, like 100%. You need to be able to do that. So there, you just made it 10. You're welcome. No excuses on this crowd. It's there. It looks like that. Label the thing. All right, other things that are 100% there are the structure hierarchy things. I'm not asking you to put them in order, but you need to know what connective tissue wrappings go around what elements of the skeletal muscle. So our connective tissue wrappings are non-contractile elements. You should know that the endomysium surrounds each fiber. The paramysium surrounds each fascicle, and the epimysium surrounds each muscle. Connective tissue wrappings surrounding each of these sub-elements of the skeletal muscle. So you should know that a fascicle is a bundle of fibers, and a muscle is a bundle of fascicles. In this case, like, this is just flat out knowing what those things are. There's some other ones where you just need to know what those words mean. Like, you should know that the sarcolemma is the cell membrane for muscle fibers. You should recall functions. of skeletal muscle. Movement. And in that movement, like breathing, the production of body temperature, control of openings in and out of the body. The muscle, however, doesn't store anything. Like, it's not a fuel source for you. It's not storing minerals like the bones did. It's not storing fat like you saw in bone marrow or adipose tissue. So, more likely than not, it will say which one of these is not a function of skeletal muscle. Those are all functions, so don't pick one of those. Openings. In and out. Now the majority of your exam is focused on skeletal muscle. But we're going to talk about cardiac muscle and smooth muscle. In terms of cardiac muscle and smooth muscle, what I'm really looking for is the comparison between those two muscle types and skeletal muscle. So we'll sort of chart this out. All right. Skeletal muscle is the only one of the three that is voluntary. It's controlled by the somatic motor branch of the nervous system. Cardiac muscle and smooth muscle are both involuntary. And while often they sort of manage themselves and the generation of contraction is sort of built into the muscle, oversight is from the autonomic nervous system, which we'll talk about when we get to the nervous system. Skeletal muscle is striated, which means we have sarcomeres. Cardiac muscle also striated, so we still have sarcomeres. Smooth muscle not striated. No sarcomeres for smooth muscle. The skeletal muscle, calcium, binds to troponin. In cardiac muscle, calcium binds to troponin. In smooth muscle, calcium binds to tropomyosin, not tropomyosin, calmodulin. Here we have all three non-contractile elements. The endomysium, the paramysium, the epimysium, all of those are present for skeletal muscle. In cardiac muscle, we only have endomysium. And here we also only have endomysium. We get T tubules here. We get T tubules here. No T tubules for smooth muscle. In skeletal muscle, no gap junctions. Cardiac muscle, we have gap junctions. Smooth muscle, we have gap junctions. Remember that in cardiac muscle and smooth muscle, the muscle cells are working together, right? Depolarization travels as a wave through these muscle tissues. That's not how that works in skeletal muscle. Depolarization is on an individual cell basis, not as a big group. Now, I'm not concerned with a lot of the detail about cardiac muscle and smooth muscle so much as I am concerned with that comparison to skeletal muscle. There's a chart at the very end of the smooth muscle, cardiac muscle lecture that has all of that charted out in some degree of detail. That's really what questions under exam are focused on, are how they differ.
What does it say, all three non-what?
The non-contractile things. All right, as we're talking about those non-contractile elements, you should know that dystrophin is this protein complex that anchors those non-contractile elements to the sarcomere inside the muscle fiber. So that they move along with the muscle. We did talk about some pathology here. Duchenne muscular dystrophy. Duchenne muscular dystrophy is when that dystrophin protein complex is not formed correctly, so they're not anchored correctly, or you get progressive death of the muscle tissue. Things to remember about Duchenne muscular dystrophy is it's an X-linked recessive disease, so only males will be affected. We diagnosed fairly early in life, lifespan not particularly long. You do need to know properties of muscle tissue. Muscle tissue is contractile, right? It gets shorter. It's conductive. The action potential travels along the cell membrane. It's extensible. You can stretch it out. It's elastic. You stretch it out, it'll bounce back to where it came from. And it's excitable. It responds to a stimulus, that stimulus being the action potential. So those are your 5 characteristics of all muscle tissue. You do need to know those. So now let's apply some stuff. And some ideas. what you can expect here. So, anytime we see questions about pathophysiology, I'm not worried about you naming diseases. At no point are you going to have to name what the disease is. You're not diagnosing things. I'll give you that and details that you need. What I care about you understanding is the mechanism that's underlying that, or being able to predict what happens if I tell you the mechanism. So a good example of this is myasthenia gravis. Now, myasthenia gravis is an autoimmune disease that damages the acetylcholine receptor at the neuromuscular junction. All right, so what we're getting here is our neuron and our acetylcholine receptors here, and we're going to take some of these out of play. What can you expect to see from this? If I'm eliminating acetylcholine receptors, I'm not like blanket taking them out of play, just some of them. What's the effect going to be on the muscle side of the synapse? It doesn't want to contract. Now you've still got some of them there. Remember what muscle tone is, right? Muscle tone is this tension that's always in the muscles, right? Your muscles always have some tension. Think your facial muscles, right? There's always some tension there. And if there's not, the facial muscles will sag. If you've ever seen anybody with Bell's palsy where half their face is paralyzed, there's no tension in the muscle anymore, and so like the face sags, and the eyelid sags, and the mouth droops, because that muscle tone's gone. Here, we've eliminated some of the receptors, but not all of them. As acetylcholine binds to its receptor, sodium's coming in, and we're starting to depolarize that cell. The more acetylcholine that binds, the faster that happens. The more acetylcholine you can get to bind, then the faster that contraction happens. So what we're doing, in essence, is reducing the amount of acetylcholine that binds to receptors. This is going to decrease muscle tone. So our symptoms here: decreased muscle tone, muscle weakness, the muscles have a more difficult time because there's not enough acetylcholine binding. We can't put those receptors back, right? I mean, the muscle might regenerate those, and you'll have periods of being symptomatic and asymptomatic with this, like you do lots of autoimmune diseases, but we can treat the symptoms. And the way we treat the symptoms is, if there are fewer receptors, we can fight that by increasing the muscle tone. And if we want to increase the muscle tone, we put more acetylcholine out there. So treatment for this is an acetylcholinesterase inhibitor. An acetylcholinesterase inhibitor increases muscle tone. Now, you don't want to force the muscles to contract by any means, or use such a strong one that like the muscles spasm out of control. You just want to increase the muscle tone so that you can reduce the amount of symptoms that we see. this sort of give and take in the neuromuscular junction is important. Because when we upset the balance that's there, when there's additional muscle tone or not enough muscle tone, we can treat that. Acetylcholinesterase inhibitors increase muscle tone. And in some forms, this is really dangerous and will kill you. So there are a couple of weapons of mass destruction that are organophosphates. One of them is a gas called sarin. This is an extreme acetylcholinesterase inhibitor. When it's inhaled, you get widespread, uncontrolled muscle contraction. This includes the diaphragm. Death is usually less than a minute. But we do have cases where people are exposed to this in smaller amounts. There were reports of this when we were in the Middle East, that occasionally something would happen in this gas. You'd have people that were exposed to it. Syria, there were people exposed to it. And they're exposed to smaller amounts where they didn't just get a ton of it and die quickly. They get some of it. So then what you see are difficulty breathing and spasms of the muscle. But we can treat that to some degree and sort of fight the effects. So an acetylcholinesterase inhibitor is increasing muscle tone because it's increasing the amount of acetylcholine in the synapse. If there's too much acetylcholine in the synapse and your muscles don't want to relax, we can put the enzyme back, but we can block the acetylcholine. So here, your treatment is to block acetylcholine. Like atropine. So the same thing that you would do with somebody that was exposed to an organophosphate pesticide locally. Difficulty breathing, the muscle spasms. You treat this with atropine and you block that acetylcholine. Now, by no means am I expecting you to remember the names of like pharmaceuticals or toxins here. The idea is this sort of balance that occurs at the neuromuscular junction, and that you understand that increasing the amount of acetylcholine in the synapse increases muscle tone or causes contraction, and that decreasing the amount of acetylcholine there decreases muscle tone. Anything that prevents acetylcholine from binding inhibits contraction. So let's think about this. Things that we could do to prevent acetylcholine from binding. So again, we've got a neuron, motor end plate, acetylcholine receptors here. For acetylcholine to come out, you've got to get calcium coming in, acetylcholine going out. And then acetylcholine binds to its receptors. So stuff that prevents acetylcholine from binding. The obvious one is block the receptors. That's an antagonist. And there are a variety of acetylcholine antagonists. Again, I'm not worried about you knowing the names of toxins or anything, but there's a lot of them. And we'll talk about different types of acetylcholine antagonists when we get to the nervous system, as we talk about acetylcholine's role at the autonomic synaptic terminals there. So, blocking receptors. Block those acetylcholine receptors would block contraction. We talked about curare as an acetylcholine antagonist. It binds in the place of those receptors, and the result of that is paralysis. We could eliminate the receptors, like we do in myasthenia gravis. There's no receptors to bind to. If the acetylcholine can't bind to its receptor, regardless of the source of that, you're going to lose the ability to make the muscle contract or decrease muscle tone. However you want to think of this, we're inhibiting contraction because acetylcholine can't bind to its receptor. We could prevent the release of acetylcholine. Remember that what triggers the release of acetylcholine is this, that calcium coming in. When the action potential arrives at the synaptic bulb, it opens voltage-gated calcium channels. Calcium enters synaptic bulb, causing the release of acetylcholine. So, if we can stop that from happening, if you can block calcium from coming into the synaptic bulb, you can stop the release of acetylcholine. And this is going to interfere with other neurotransmitters, so this is not something that's super great to do. But again, I'm not worried about like specific pharmacology here. I'm concerned with this idea, preventing the release of acetylcholine preventing acetylcholine from binding. And that's just another way we can do this. Anything that does that, if we stop acetylcholine from binding, we stop contraction. On the other end of that spectrum, anything that causes more acetylcholine to bind amplifies contraction or increases muscle tone. Things that we can do to cause more acetylcholine to bind. Release more acetylcholine. That was easy. The more acetylcholine that's out there, the more acetylcholine can bind. The other thing, inhibit acetylcholinesterase. If we stop that enzyme from breaking down acetylcholine, there's more acetylcholine out there. So you either have something that causes more acetylcholine to be released, or you have something that stops acetylcholinesterase, and either way, the result is going to be increased muscle tone, muscle contraction. So on your exam, when you see questions about this that mention anything about specific like cholinesterase inhibitors or acetylcholine agonists or anything like that, don't get lost in that idea. Don't look at that and think, I have no idea what that means. That's not the point. The point is this big picture idea. If I tell you there's something that's, a competitive reversible antagonist of acetylcholine, the only part of that that's important is that it's an acetylcholine antagonist. It's blocking that receptor. That's where you need to sort of hone in on. That idea being that it's going to inhibit contraction. Don't worry about like the name of whatever it is. The mechanism is what's important. For this exam, more so than the prior two exams, your ability to synthesize this together and apply it to some sort of clinical situation is becoming more important. This will be true in the nervous system as well, and pretty much solidly through AMP2. Now, I'm not going to ask you step by step what's happening to this, what comes after this step, or what happens after acetylcholine binds, or what happens when the action potential travels down the sarcolemma. I'm not worried about that step by step. The big picture is the important thing. Action potential travels down the sarcolemma. Calcium comes out of the sarcoplasmic reticulum. One thing that you do need to know, as at the final trigger for contraction, is always the increase in intracellular calcium. That calcium comes out and it binds to troponin. Remove tropomyosin, we start that sliding filament theory. But it's that calcium that's triggering that movement. The sliding filaments. Things that you need to know about our sliding filaments. In skeletal muscle, as we mentioned, calcium binds to, that's our sort of regulatory thing going on here. Troponin moves tropomyosin. You need to know that tropomyosin blocks the binding sites on actin. I want to tell you that like 100% that question's on there. Which of these proteins blocks the binding site on actin is tropomyosin. You're welcome. You're like a 12 now. All right. Troponin is tropomyosin, myosin binds to actin. You need to know that ATP breaks the myosin actin cross bridge. And the energy from that ATP is what's providing the energy for movement. This ATP is broken down by an ATPase into an ADP and that inorganic phosphate, and there's energy released from that. We use the energy released from breaking that chemical bond to make the movement happen. This ATPase and skeletal muscle is in the myosin head. Again, that question's full on there. It's like if you were looking for ATPase, enzymes, and skeletal muscle, where would you find them? It's in the myosin head. ATP breaks the cross bridge. One thing that we looked at as we talked about the idea of ATP breaking the cross bridge is the idea of rigor mortis. When there's no ATP to break the cross bridge, but that still forms. Myosin binds to actin, but there's nothing left because you're dead. When you're dead, decomposition sets in very quickly. And that sarcoplasmic reticulum, which stores calcium, falls apart. Calcium leaks out. Troponin binds to calcium. You lose tropomyosin, myosin binds to actin. Then we stop because there's no ATP, so the cycle can't keep going. And we have rigor mortis. Okay. So now, as we're talking about ATP, we need to talk about the way that skeletal muscle makes ATP. You'll notice I've limited us here to skeletal muscle. That's because smooth muscle and cardiac muscle, especially cardiac muscle, just use aerobic respiration. Smooth muscle, that's not so much of a concern, but cardiac muscle, only aerobic respiration. And I should write that down because that's probably on there too. All right, so skeletal muscle ATP production. We've got aerobic and anaerobic methods of making ATP. Immediate energy will either be from stored ATP, and we can use the oxygen stored in myoglobin. Myoglobin red pigment stores oxygen. That buys enough time for our phosphagen system to kick in. Phosphagen system, got the two enzymes, myokinase, creatine kinase. This uses that direct phosphorylation where we take an ADP and give it a phosphate, so we've got some ATP. This buys you a little more time. Then we have anaerobic glycolysis with a glycogen lactic acid thing that's going to happen here, where we're going to take glucose or a glycogen, and you get like 2 ATP and lactic acid. All of this to buy enough time so that we get back to aerobic respiration. We're taking oxygen and glucose, and like 36 ATP, water, carbon dioxide. So your skeletal muscle is really good at this, and we'll use any of these things depending on the demand placed on the muscles to make ATP. we applied this to different activities that we do and the limiting factors on those activities. So for glycolysis, our limiting factor here is glucose or glycogen. The carbohydrate is what's limiting us. It's not, glycolysis is not super efficient here without oxygen in play, and so we have to use a lot of it. Right, you're using a molecule of glucose, you're getting 2 ATP. So that means you can run through it faster. So the limiting factor there is that carbohydrate. So when you have athletes that are in these sort of anaerobic events where they're going with these short bursts of activity and then stopping, they'll load up with carbohydrates, even during the event. Right? They're bringing Gatorade and sugar water so they can fuel this reaction. For aerobic respiration, oxygen is the limiting factor. Because you can burn glucose, you can burn glycogen, you can burn fat, you got the fuel. You're only limited by how much oxygen you can bring in and how much of this reaction will take place. One of the things that we talked about here was what we see often in endurance athletes who are cheating, and that's blood doping. Blood doping is increasing the amount of oxygen that the blood will carry. Put more oxygen there in the bloodstream, then you can give more oxygen to the cells. Incidentally, aerobic respiration takes place inside the mitochondria. And then your mitochondria can make more ATP. Now, is this gonna work for like me right now? Like I'm gonna like shoot up an EPO and then run down the hall? That's not happening. Not the EPO part, the running. Because those mitochondria are built up with training. So this is not like a magic trick where like you do this and then now you're an athlete. You're an athlete and you do this to get an advantage. Because your muscle cells adapt to what you do with them. If you're doing these aerobic events, then your muscle cells get better at aerobic respiration. They make more mitochondria. They store more myoglobin. They get better at those endurance activities. So then the only advantage you can give yourself is to somehow get them more oxygen to go through. Incidentally, on that note, if more oxygen in the blood is a performance advantage, why don't they just breathe a higher, like, why aren't they doing like the stupid little oxygen tanks you buy at CVS or whatever? Have you seen these? They're like $40, man. And it's like this can, it's like Spaceballs, where it's with the canned air, and it's got a little mask on it, and you breathe in a higher percentage of oxygen. Why not do that? Even right now, at the percentage of oxygen that's in the atmosphere, which is about 21%, your blood is about 99% saturated with oxygen. So you want to breathe 100% oxygen, there's absolutely no benefit to it right now, assuming that you're not struggling to breathe. We only do that when somebody has issues, breathing issues, respiratory issues, where we can't get enough oxygen in their blood through normal breathing. But normal breathing, your blood's pretty well saturated. So the only thing we can do is put something in the blood so they can carry more. So you can shoot yourself up with your own blood, or use hormones that make you make more red blood cells, so you can carry more oxygen in the blood. But adding more oxygen to what you're breathing gives you no advantage. In terms of aerobic and, or I should say, like endurance and resistance exercise, the changes there are important. Understanding that your muscles adapt to what you're doing to them. Right, in resistance exercise, the muscle fibers start to look more like those type 2 fibers, and as I mentioned that, you do need to know the difference in the muscle fiber types. We have our type 1 fiber. These are red fibers. They store a lot of myoglobin. That's why they're red. They are slow oxidative fibers. So they like to use aerobic respiration. These are high endurance fibers. They're also the smallest fibers. Our type 2 fibers are white fibers. They're white because they store a lot of glycogen. These are the fast glycolytic fibers because they use anaerobic glycolysis. These are high fatigue fibers, but they're the biggest fibers. And then in between the two, you have your intermediate fibers. Intermediate fibers are fast oxidative. And they're really in the middle, right? There's some glycogen, there's some myoglobin. They can do the aerobic thing really well, they can do the anaerobic thing really well. And their fatigue somewhere in the middle, the size is somewhere in the middle. They're intermediate all the way through. And any given muscle is going to be a blend of the different fiber types. But some muscles will be predominantly more one or the other. So for instance, the postural muscles that run up and down your spine, those are predominantly type 1 fibers in those muscles. They're not very big, so they're not very strong, but they're good at endurance. They don't have to be very strong and very big to do their job. Their job is just to hold you upright. Type 2 fibers will find like the muscles of the thigh and the arm, where we get the capability of these short bursts of like a powerful activity. And then intermediate fibers somewhere in between. With training, we can shift these back and forth. Like with regular endurance exercise, those type 2 fibers start to look more like intermediate fibers. Those intermediate fibers start to look more like type 1 fibers. We kind of shift this way. With regular like resistance exercise where you're trying to build muscle, like you sort of go the other way, and the muscles get better at what you're doing. They adapt to the load that you're placing on them.
So you're saying that the intermediate fibers can...
Yeah, they'll shift over. We talked about different types of muscle contraction. We have isometric and isotonic contraction. Isometric is contraction with constant length. The muscle fibers aren't getting any shorter. We're just increasing tension. Right? Try to pick up something that's ridiculously heavy. Your car. You're not going to bench press your car. You can try really hard. All you're going to do is build tension in the muscles, right? You're pushing up as hard as you possibly can. The muscles aren't changing length. All that's happening is tension is increasing. That's isometric contraction. Just the increase in tension, but no change in length. Isotonic contraction. Contraction with constant tension. And there are two types of isotonic contractions. Concentric and eccentric. Concentric isotonic contraction, the muscle fibers get shorter. Right? Flexion of the forearm. Muscle fibers get shorter. Eccentric contraction is contraction as the fibers get longer. Now, contraction definitely means, right, shortening of the fiber. Those filaments are linking together. But the blending of the concentric and eccentric contraction is what creates the smoothness of motion for us. So that when you have flexion of the forearm, you can feel that tension in your bicep muscle. But when you extend your forearm, you still feel tension in that muscle until you get back to the stopping point. You're blending that together so the muscle's not just like popping back like it's an elastic, like a rubber band. Right? It's maintaining that latch state off and on so that you have that smoothness of movement. That's the eccentric contraction. Those fibers are latching and letting go and latching and letting go as the fiber is getting longer and relaxing. We talked about different fascicle arrangements. Circular, parallel, pinnates. This is in order from the weakest arrangement to the strongest arrangement. Those fibers can put a lot more force on its point of attachment when they're all attached at the same point. Circular muscle, that arrangement's not particularly strong. And we use those circular muscles to open and close like openings, like the orbicularis muscles around your mouth and around your eye. And there are other muscles throughout the system, like various sphincters throughout the digestive tract and the urinary tract. We have these circular muscle arrangements. Parallel muscles, arranged parallel, not particularly strong, like the postural muscles that go up and down your spine. Adductor muscles that move your leg back in, those sorts of things. Pinnate muscles, those big muscles, the quadriceps and the hamstrings and the gluteal muscles, and the biceps and the triceps, you'll see them all run to a point. Now, as we have these isotonic, isometric contractions, and we're talking about varying the length of the fiber, remember that to vary the length of the muscle fiber, we're changing the frequency of the action potential, of the stimulus from the nervous system. So that as we increase frequency, of the stimulus, that is your action potential, you're increasing tension in a fiber. This is temporal summation. Those action potentials, the faster they fire, the shorter that fiber gets. This is the sort of coding that we see. This is how your brain tells your muscles how much to contract. The difference between, having your forearm fully in flexion and fully in extension and being somewhere in between. That's coded for, and how fast that muscle fiber is being stimulated by the nervous system, how fast that action potential arrives. The faster it arrives, the faster you see that action potential, the shorter the muscle fiber gets. And you can keep doing that. Like, you can keep firing faster, and you can keep building tension in that muscle. Even if you're not creating movement, you can keep building tension in the fiber to a certain point. That point where you can't get any more tension in the fiber, it's as much as it'll go. We get that sustained contraction. That's tetany of the muscle. Technically, this is called incomplete tetanus, and it's sort of a wave of contraction. You see it sort of fluctuating back and forth on a readout. We don't do complete tetanus because it's not something your brain can't send the signal fast enough to get complete tetanus, where it's just a fused contraction. There's always going to be some latching and letting go. So this is temporal summation. The faster you fire the action potential, the shorter that muscle fiber gets. If you want to use more muscle fibers, then your brain sends more signal. That's multiple motor unit summation. So back to definitions, you should know that a motor unit is a single neuron. And all the fibers it controls. And multiple motor unit summations, which is also called recruitment, is when we increase the intensity of the stimulus from the brain to fire more fibers. Your brain says it's time to use some more muscle. The muscle that we're currently applying to this job is not doing it. Let's recruit some friends. This isn't a conscious decision on your part. You use more muscle to lift heavier objects. To do more work, it's going to require more muscle, and your brain does this for you. It recruits more muscle fibers as necessary. Now, at any given point, we're probably not using more than about a third of the muscle fibers in the muscle simultaneously. We're taking turns, but we're recruiting those fibers. Our order of recruitment, we bring the smallest fibers in first. So those type 1 fibers are the first recruited, and then we bring in intermediate fibers. That's not enough, we can bring in those type 2 fibers. We're going to start with those type 2 fibers. This is an intense stimulus from the brain. So we get this short burst of powerful activity, and we start with that. Multiple motor unit summation is responsible for like that phenomenon of like people being able to lift cars off themselves or large objects off babies or whatever. The brain is like, well, let's give it a shot, and fires all the muscle fibers at once. And it either works or you die. Now, remember that as a byproduct of muscle contraction, we're also getting heat. This is the principle behind shivering, right? Shivering is skeletal muscle contraction. Your brain takes over for you and causes this sort of involuntary skeletal muscle contraction. But as the muscles contract and relax, you generate heat. Keep the muscle completely contracted and like just hold that contraction, it's still going to be generating heat. This is one of the issues we see with the infection we call tetanus. So the infection we call tetanus is from a bacteria, Clostridium tetani, two eyes. And this causes the tetanus infection. The major symptom of this is tetanus, tetany of the muscles. The muscles contract and stay there and as you have that sustained contraction, we get the production of heat. It'll cook you. Hyperthermia is a major concern in patients that have tetanus. You shouldn't get tetanus because you know there's a vaccine that's been available forever and is widely used and super effective. You get tetanus from anything that breaks the skin. I know it's always that whole rusty nail thing. It has nothing to do with the nail and nothing to do with the rust. It just happens to be something dirty that breaks your skin. If you get bitten by something and you haven't had a tetanus shot in the last 10 years, you get a tetanus shot, especially if it's a person, a cat, or a snake. The last tetanus shot I had to get because the snake down the hall bit me. Hate that snake. Still, I'm bitter. Everything wouldn't let go. Like 30 minutes of that thing attached to my hand. And like then it was like not my snake, and I'm like, what am I gonna do? It's not my snake. I can't like hurt it. Although that was my initial instinct, was like, must kill snake. And I was like, oh crap, it probably costs money, so I probably better not do that. So then it just stayed attached to my hand for like 30 minutes, walking around the hall. I'm like, what do I do with this? And I guess finally it decided it couldn't eat my hand, so it let go. Still hate it. And then I had to go get a tetanus shot. So, you shouldn't see tetanus. Like, it's not something that comes up a lot. It comes up more often than it should, but it still shouldn't come up a lot. But that's sustained contraction. It's also going to burn through your resources very quickly with the sustained contraction. The only patients I've ever seen that had tetanus are on feeding tubes, and they have them on thousands of calories a day, like way more than you should get, because they're burning through those calories that quickly with the muscles being contracted. And they're trying to keep them cool too, so they don't overheat. You should know that cardiac action potential has a long absolute refractory period to prevent temporal summation. Because of the way cardiac action potential works, you can't get the action potentials close enough together to get fused contraction. The heart won't like keep getting shorter when you send the signal faster. It's going to contract and relax because the refractory period is so long. Now, I'm not worried about what causes that. You don't need to know like if it's calcium influx causing the plateau in the cardiac action potential. That's a topic for another semester. But you do need to know that cardiac muscle won't do temporal summation because it has a really long refractory period. All right, let's things. All right, if I tell you to choose the organ or whatever that has cells connected by gap junctions and the same cells exhibit a stress relaxation response. What's some choices? Pick one. It's the bladder. So, connected by gap junctions, that immediately eliminates skeletal muscle. Skeletal muscle is not connected by gap junctions. Stress relaxation response, that tells you that it's smooth muscle. Which the heart is not smooth muscle. So that leaves you with the organ that's made out of smooth muscle. And not all smooth muscle exhibits a stress relaxation response, but you do see this in the hollow organs like the bladder and the stomach that are meant to store something. So if I tell you to choose the organ that has cells connected by gap junctions, I'm not going to write all this again. Use your imagination. And desmosomes, which one of them would you have picked?
Heart.
It's the heart. Again, gap junctions immediately eliminates skeletal muscle. Desmosomes, well, that's not the bladder. The heart is what we have here, connected by gap junctions. And desmosomes, those connections between cardiac muscle cells are called intercalated discs. And the heart contracts as one big unit. All right, if you have a patient with troponin in the bloodstream, which of these muscle types could possibly be damaged if you've got troponin in your bloodstream? Where do you find troponin?
In the heart, right?
In the heart and in the skeletal muscle. Both of those have troponin in them. So if you've got a patient with troponin in the blood, they've either damaged skeletal muscle or they've damaged cardiac muscle. Normally when you draw for troponin, you've got a patient that comes in with chest pains and that crushing thing and pain down the arm, all those heart attack symptoms, and you're trying to figure out if that's a heart attack. Sometimes they come in with the chest pain and it's intense and it's not very specific, and you draw for troponin to see if they're having a heart attack or whether it's acid reflux, because sometimes those look very similar. Smooth muscle, no troponin. Like all the other tissues that we talked about, muscles respond to what you do with them. Right? They adapt to the load you place on them. We talked about this in terms of the overload principle. The overload principle. To achieve any benefit from exercise, muscle has to be overloaded so it can adapt. Like you want to run a mile and you haven't, run forever. You start small and you add to that distance so the muscle can adapt to the load you're placing on it. You push it and then you keep doing that until you adapt to that distance, right? And then you're good and you can run a mile in your sleep. And that's weird if you do that. You can just, it's good, you're fine. Running miles, nothing. And then you add to that distance. And you keep adding. As you overload it, you get more benefit. The muscle adapts to the load you place on it. At the same time, if you don't do anything with the muscle, muscles atrophy fairly quickly. Like any other tissue. If you don't use it, you lose it, and the muscle is no different. And we see this in people that are paralyzed. The muscles will atrophy quickly. People that are in a coma, the muscles will atrophy quickly. If people are in a coma for a long time, often they'll come in and put those tense units on them and make the muscles contract to slow that atrophy down as much as possible. But even like being in a coma for a month, you're gonna have to go through physical therapy when you get out. Assuming you get out. Being optimistic. Because the muscles will atrophy very quickly. Let's see. Another thing we talked about here is the maximum oxygen uptake, or the Vo2 Max. This is the most oxygen your cells can use, like during exertion or exercise or whatever. I don't mean like it's because there's no more oxygen. I mean that you've only got so many mitochondria, right? And those mitochondria are working, and they can only do so much. Like if you got like 4 mitochondria, it's really exaggerated here. You got like 4 mitochondria, they can burn like 4 oxygens or whatever. If you got any more, that's a waste. This Vo2 Max increases with physical fitness, I guess you should say, training. It's much higher in endurance athletes because their cells have adapted to that endurance. They've adapted to needing to use that aerobic respiration. So mitochondria do aerobic respiration. The cells make more mitochondria. And they can burn more oxygen now because oxygen was our limiting factor. Now their cells can burn more of it. The list of topics here. Okay. Do those. Okay, I think with that we've been over everything that's there. I can't think of anything that I've left out at this time. Focus on this stuff. Some of it flat out, like we just did the questions. Quite a bit of it was just flat out what's there. Make sure that as you're going back and studying this, that you're focusing on what we've talked about today. don't believe I've left anything out, it's all there, so there shouldn't be any surprises there. If you have any questions, you study, just let me know. Otherwise, that's it. Take a break, and you can have the rest of the time in the lab with the structures on the table, the models that are laid out. The rest of the time is yours.